All right, so thank you for giving me the opportunity to discuss some of the work we did on AAVs and mass analysis by CDMS and mass photometry. So yeah, let's get started. First, a little bit of background on the project. So adeno-associated viruses, the AAVs, they became quite popular as a vehicle for gene therapy in recent years. So here I put up several therapies that have been approved by the FDA and or EMA that contain AAVs. And last year, or two therapies could be added. Those are AAV-based therapies that target bleeding disorders, basically, so hemophilia A and hemophilia B, and they contain AAVs that basically express coagulation factors, so coagulation factor 9 and coagulation factor 8. So it's really exciting to see that. Of course, there are many clinical trials also ongoing or planned, so perhaps in the future even more therapies can be added to this list. Seems very promising. So why use AAVs? Well, AAVs have several advantages. They are have a very low immunogenicity. They are non-pathogenic, so they cannot replicate themselves. They need additional viruses in order to replicate, such as the adenovirus. And they have a long-term expression of their transgene, and they have a very broad tropism. So AAVs can, in principle, target many different tissues. There are many different variants of AAVs. So there are some serotypes which are well known, but even beyond that, there are many other variants. And of course, at the moment, there's quite some research going on in order to engineer the capsids in such a way that even different tissues can be better targeted and more specifically targeted. An AAV capsid is quite small, actually. It's about 25 nanometers in diameter, and this is protein capsid, and then can package a single-stranded DNA of about the maximum capacity of 5 kilobases. Of course, in setting up a gene therapy with the AAVs, an important part is the production of the AAVs themselves. So just for now, for this talk, I will quickly discuss two of the main production platforms that can be used. Basically, a mammalian-based expression system or an insect cell-based expression system. And if you would use a mammalian-based expression system, you can use HEK cells or HeLa cells, and they then need to be transfected with different plasmids in order to produce the AAVs. So one plasmid containing the transgene, another plasmid that contains rep and cap genes. So these are proteins that can actually form the capsid and then fill it with the single-stranded DNA of the transgene. And next to that, you might need a plasmid for helper factors in order to get the replication of AAVs going. Because as I just mentioned, AAVs cannot replicate themselves. They need additional factors. Next to the mammalian expression system, you could also use an insect cell-based expression system. So you're basically using insect cells, SF9 cells, for instance, and then you would use baculoviruses. So these are viruses that are specifically infecting insect cells, and you would, for instance, use one baculovirus that contains a transgene, and then a second baculovirus that contains these rep and cap genes. In this case, you don't need to worry about any additional helper factors because the baculovirus itself already provides this function for the AAVs in order to replicate the AAVs. All right, so following production of the AAVs, of course, an important question though. An important question is how much after making the AAVs actually contain a transgene, contain a genome, and are therefore useful as a gene therapy vehicle. Um, Unfortunately, if you use either one of these expression systems in order to create recombinant AAVs, only about 5 to 30% of these AAVs actually contain a full genome. And this can also vary a lot between production runs and, of course, between different production system you use. So it's important to following the making of your AAVs to assess how much of them actually contain a transgene. So in the past, several assays has been set up and used in order to answer this question. For instance, qPCR or optical density, and ELISA assays, also dynamic light scattering and electron microscopy all have been used in order to see how many of your AAVs actually contain transgene. Fortunately, these techniques usually answer just that, like whether it contains single-stranded DNA or not. but not if there will be any partial filling of a capsid, unfortunately. And also these techniques are quite laborious, so and it takes some time, so it's not really fit for making it in a high throughput manner. And there could be potential biases as well. For instance, if you do electron microscopy and you have some staining artifacts, you might think capsids are filled while they're actually empty. Okay, so at the moment, the gold standard for assessing this question is using ultracentrifugation, so analytical ultracentrifugation. And thereby you can nicely separate your empty particles and your filled particles, and you can even distinguish some particles that might be partially filled, so only contains half or some part of the transgene. But unfortunately, this technique requires quite some material, and also a lot of time. Is quite laborious, so it's not really fit for a high throughput method. So basically, that's where our lab came in, and we thought, okay, we're a lab that's focused mainly on mass spectrometry, and can we use mass spectrometry in order to determine whether AAVs characterize AAVs and see how well they are packaged. Basically, answering the question, can we determine the molecular weight of our AAVs if we have a pool of AAVs? Mass spectrometry is quite sensitive, so in principle you need a little amount of material. Also, we would just directly infuse the AAVs in a mass spectrometer, so no liquid chromatography steps and such. So you would, in principle, use only a little amount of material. It can be quite fast also for this reason. So it might be that there's a potential there also to use this as a high throughput method. Okay, so if we have our mass spectrometer in our lab, so we usually use a Orbitrap-based mass spectrometer. So the mass analyzer is a small Orbitrap. If we take the machine in the lab, and then of course it's nicely designed and it's got this outer shell and all the electronics, but if you take all that away and just look at the main part of the machine, then most of the parts will just are there to focus and guide the analytes all the way up to the mass analyzer here, which is the small Orbitrap element. And these Orbitrap-based mass spectrometers are usually used for small molecules and peptides, but here we actually want to try to see if we can measure complete intact AAVs and determine the molecular weight of our AAVs. Okay, so if we have for instance, a relatively small protein, IgG, about 150 kilodalton, how will we measure such an intact protein with our Orbitrap mass spectrometer? Well, first we have to put our protein in a solvent that is quite volatile, so for instance, ammonium acetate, and then go from solution towards the gas phase in order to then bring the IgGs in the vacuum of the mass spectrometer all the way up to the Orbitrap mass analyzer. So if we have our IgGs in solution, we would then put an electric current on the solution and place it in an electric field. So droplets will be formed containing our IgGs, and on the outside there will be charges. And as the droplets become more and more smaller because of the solvent, it will start to evaporate. And in the end, we only end up with just single antibodies, single IgGs, which on the outside contains charges. And then it goes into the vacuum of the mass spectrometer and is guided nicely all the way up to the Orbitrap mass analyzer. And there they will start to spin around the electrode in the Orbitrap. And this spinning will give us then a mass spectrum like here. So this is a spectrum with, on the x-axis, we have the mass over charge ratio. And then here we have the intensity of all the IgGs that are spinning in the Orbitrap. It's just important to note, perhaps, that while going from solution towards the gas phase in this electrospray all the way up to the mass spectrometer, into the vacuum to the mass analyzer, the protein stays folded and intact. It stays in its native conformation. So basically, that's why this is also called native mass spectrometry. And when we have our mass spec here, spectrum, you can see that we have IgGs that are measured, and they all can carry different amounts of charges. Some carry 25, some carry 26, others carry 24. And with this spectrum, you can say, okay, well, got our M over Z ratio. We have the charges here. So we can calculate for each one of these the mass, and then if you just multiply the charge by the MOZ and then subtract the charges again, we get a mass of around 150 kilodalton. So that's good. So it's important to be able to distinguish the different charge states here in order to calculate properly the mass. Okay, so let's go further, a bit bigger protein. It's a protein complex, actually, GOEL, chaperone from the E. coli. And it's a complex, so it has different subunits that are non-covalently associated. And it's about 800 kilodaltons. Can we also measure this? Yes, so if we use GOEL, so that can nicely spray into the mass spectrometer and be analyzed in the orb trap. And we get a mass spectrum right here. So it carries a little bit more charges because it's also bigger. And also from here you can calculate back mass, which is about 800 kilo. If we then go further, again a bit bigger, IgM complex. So this is a pentamer of IgM antibodies. So these are 5 IgM antibodies together. With 1 megadalton, again a bit bigger. So what you'll see, if we also measure this in the mass spectrometer, carries a bit more charges. But also here nicely you can see the different charge states and then calculate the mass from these different charge states, which is close to 1 megadalton. Okay, so then the question, of course, can we even go further and actually measure intact virus particles? So for that we tried different virus particles. This is the dodecahedron, which is like a base part of the adenovirus, the cowpea chlorotic mottle virus, which is a bit bigger, so 4.5 megadalton, or the flockhouse virus, which is really a big viral particle, about 10 megadaltons. And the idea was, okay, can we also see these viral particles in our mass spectrometer? And for that, we probably have to go even further in our detection range, in our MOZ range here, even beyond the 20,000, which was thought to be really a bit of a limit. But following optimization of our mass spectrometer, so changing a bit the voltages and the pressures in the machine in order to guide these big viral particles properly to the orbitrap. Indeed, after doing this, we can nicely measure also these viral particles. So here again, the dodecahedron is 3.5 megadaltons, the CCMV 4.5 megadaltons, and even the flockhouse virus, which is about 9 to 10 megadaltons, and it's all over close to the 40,000 MOZ. Okay, so of course the question next is, can we also measure AAVs? Well, yes, also that works quite nicely. The AAVs, we could spray them in the mass spectrometer and nicely guide them to the orbitrap and measure there the different particles. However, if you look at the spectra itself, it's quite messy. You do not see nicely these clear charge states as we saw earlier for the other virus particles. It's like a complex mixture of peaks, and it's quite a messy signal. So why can we not nicely see these charge states, which we saw before in the other ones? Why for the AAVs this is not working? And from this also we cannot calculate any mass in the end. Well, actually in hindsight that kind of makes sense, because if we looked at the previous particles, they all had a isoform subunit. So they had like one protein subunit, which was the same mass for the whole capsid. For the dodecahedron, we had this penton base. The CCMV had a capsid protein, which was just single capsid protein for the whole capsid. And also in the flockhouse virus, there's this alpha protein, which just has a single mass for the whole capsid. However, for AAVs, it's different. The other capsid consists out of 3 different proteins with different sizes. So we have VP1, VP2, VP3, and they all have 3, all of them have a different size. So VP1 is about 82 kilodalton, VP2 66, and VP3 around 60 kilodaltons. And a capsid contains 60 capsid proteins total, and it can be a mixture of VP1, VP2, VP3. And what could happen, for instance, if you would have one AAV particle that contains 1 VP1, 9 VP2s, 50 VP3s, you would get a spectrum like this. But if you have another particle which contains 1 VP1, 10 VP2s, and 49 VP3s, the spectrum would be slightly shifted. And if you even have a third one with 1 VP1, 11 VP2s, and even less VP3s, 148, it will be even further shifted. And you would measure all these different AAVs at the same time in your Orbitrap, so you get this complex mixture of signals, of spectra basically, and this congested spectrum is really tough to then disentangle again the mass from, if not impossible. So this is basically the mass heterogeneity, which is really a problem of the AAVs, which makes it very difficult to use native mass spectrometry in order to then measure the mass of our particles, of our AAV particles. And so native mass spectrometry work nicely for our particles we saw earlier, like GROEL or the IgM or our different viral particles. But unfortunately, because of the mass heterogeneity in AAVs, we get these congested spectra, and it's really not possible to get any mass out of those data. So instead of measuring all these particles in the Orbitrap at once, spinning around the electrode, can we not instead measure these AAV particles, for instance, one after another, and then we do not have to worry about these interfering spectra and this mass heterogeneity. So basically, we now have two methods that use the single molecule approach in order to get a mass from the AAVs. And I will just explain both of these techniques, the Orbitrap-based charge detection method, a little bit going forward in the previous story, and a completely different method using light and microscopy. So basically, that's the mass photometry. So I will just show both these techniques and then say how we use it in order to get a mass from our AAV samples. All right, so first the Orbitrap charge detection mass spectrometry. Is it possible to actually measure just a few particles or a single particle in the Orbitrap at a time? So basically, yes, it's possible to measure a few particles in the Orbitrap, for instance, GROEL, if you would have used a very low concentrated sample, and you would down-tune the machine basically in order to allow only a few GROEL particles at a time in the Orbitrap, you get a spectrum like this. But now you see these small peaks, basically like spikes, which every peak is a single GROEL particle that is spinning in the Orbitrap and gives a signal. So it's possible to measure just a single particle, but how do we then get a mass from such a spectra? All right, so therefore we look at the data a little bit differently. We can make a 2D histogram, for instance, where we put on the x-axis the M over Z again, the y-axis also the intensity, but now we just place a dot for just this single GROEL signal that we detected, and we place the dots for all the other GROEL signals as well, and we just repeat many times these measurements, and then you get an image like this, like here. So now we basically have a 2D version of what we saw earlier in our native mass spectrum, where we saw this nice charge distribution. Also here we can see these different charge states, but now this has been constructed just by measuring one GROEL after another and placing them in this histogram. So you can also imagine that this is actually quite sensitive way of working, because you only need a few well, thousands basically of particles in order to fill up this histogram, and the measurement would take about like 10 to 30 minutes, but you only need a very low concentrated sample and a little bit of material. Okay, so you can see here nicely these charge states again that were also visible in the native spectrum, and you might also observe that if you draw a line, you can see that there's like a linear scaling of the amount of charges and the intensity that is measured for each of these GROEL particles. So basically, the more charges that are there on a GROEL particle, the intensity is higher. And you can place this also a little bit differently in a different plot. So if you would now put the charges on the x-axis and the intensity on the y-axis, then again we can place our GROEL measurements here, and we can repeat this assay, but then for all different kinds of protein complexes. So again, if we would do the same, but then for antibodies, which show to have much larger, less amount of charges, they would end up here on this part of the graph. And if you, for instance, take again the blockhouse virus, which was much bigger and much more charges, they would end up on this side of the graph. And we can see again that there's like a linear relationship between the charges and the intensity that is measured for such particles. And from this line, we can also get a slope. And then we would say, okay, every charge brings about 12.5 units of intensity when it's measured in the orbit trap. And basically, when we have the intensity, for instance, here in our spectra, we can then calculate back the charges that is on each of these Coriell peaks that were spinning in the orbit trap. And also, when we have the charge and we have the m over z, we can also basically calculate for each individual Coriell particle, again, the mass by just multiplying the charge with the m over z, and then subtract the charge again, and then we can place these masses in a sort of mass histogram like here. And we could see that in the average of all these Coriell masses we now measure, it's again about 800 kilodaltons. So there's a bit of a uncertainty in the intensity, which comes with the carrying a certain charge. So we can see that also in the width of the distribution here, there's a bit of a variety, but overall, the average mass is about 800 kilodaltons. So that seems to work nicely. How about actual AAV particles? How can we measure this using the same charge detection approach? So actually, AAVs would be ideal candidates because we only want to use a very little amount of material. So that's good. And it's, as we saw before, a very heterogeneous sample. So this would show us if we can really get a mass number out of this method. All right, so we here would use AAVs, empty ones, so they do not contain any transgene, and they're from the AAV8 serotype, and we actually got them from Serion. So let's see. Yes, so basically we would take the same approach, and we can measure here the single AAV particles, as we saw for Coriell, these spikes, these small peaks in our mass spectrum. If we then plot it in this 2D histogram, we see this cloud basically of AAV particles, so we cannot distinguish anymore these nice charge states as we saw earlier, which makes sense because of the mass heterogeneity. But we can, for each individual AAV particle, convert intensity to charge using the slope we saw earlier. And then with the charge and the m over z for each AAV particle, we can calculate the mass and place it in a mass histogram like here. And then we end up with a average number of about 3.8 megadalton of molecular weight for an empty AAV8 particle. So just to show, using the orbit trap based method, we can now get a mass number out of our AAV particles, about 3.8 megadalton. While if we use the previous native MS approach, it's virtually impossible to get any mass from this data. All right. Now about mass photometry, the other method we can use for single particle mass analysis. Well, mass photometry is based on a completely different principle. So it uses microscopy and laser light. And basically, if we have our AAVs in solution, PBS, we put it on a glass cover slip, and we shine a laser light on the glass cover slip, most of the laser light will be transmitted and gone, but a little bit of the light will also be reflected by the glass surface. And if an AAV then lands on the same glass surface, there will be scattering, and the scattering will interfere with the reflected light and kind of causes a small shadow, so a dip in the signal. So, for instance, if we take a protein, thyroglobulin, these are multimers, so we have a monomer, we have a dimer, trimer, tetramer. And if we do the same thing, so we put it in the buffer solution, PBS, put it on a cover slip and shine laser on it, we can detect these scattering events every time a thyroglobulin protein lands on the surface. And here's just a frame from a movie. So you would record these movies, and this is just a small frame. You can see every time when a protein lands on the surface, it gives this signal, this dip, this dot, basically, this black dot. All right, so these dots, they are basically a contrast value. And these contrast values we can put again in a histogram, like here. So if we place this in a histogram, we can see again our monomeric thyroglobulin, the dimer, the trimer, and the tetramer. And the contrast value nicely aligns or is linear with the mass of the multimers we see here. So you can plot this. We have here our mass of the different multimers, and then the contrast value, and you can see clearly this linear relationship. So also from here we can say, well, we know how to basically translate our contrast value into mass. So how about AAVs? What if we just measure AAVs? Well, we place our AAVs in PBS and also put it on a cover slip, shine laser light, and we get an image like this. Where again we see these black dots. That is every time when a AAV is landing on a surface, we have this signal. It can also be that sometimes an AAV is detached from the surface, so you see a reverse, you see this white signal. But those we disregard. We only look at the negative contrast values here, these dark spots. And then translate that contrast value into mass, and also that we can again put it in a mass histogram like here. So this is a AAV8 empty capsid from Sirion again. This time using mass photometry, we get a histogram like this, and also a mass about 3.8 meganol. So if we put them all next to each other, so this is our Orbitrap-based CDMS method, we get about 3.8 megadalton for our AAV particles. Mass photometry, again, 3.8 megadalton. And native MS, well, that doesn't give us much of an answer. Okay, if we repeat these assays several times, so this is CDMS, which we repeated, there's a bit of variation, and also mass photometry, we can repeat. Also there is a bit of some variation, a little bit more than with CDMS, but still both techniques seem to be quite reproducible. All right, so far we only have looked at AAV8 from Sirion, and Sirion uses a mammalian expression system, so these were made from HEK cells. But what if we look at AAVs, same serotype, again AAV8, and also empty, so not carrying any transgene, but this time from an insect cell expression system, so SF9. How would that look like? Okay, I repeated basically both the same assay. So here on top we see the Orbitrap CDMS measurement, and we can see a major population of our empty particles and a very small side population which carries a little bit more mass, so potentially partially filled. And the mass photometry, we actually see the same picture. So here we have main population of empty particles, and again, a small side population of particles that contain something extra. The mass here is about 3.7 megadalton for CDMS and 3.65 megadalton for the mass photometry. So if you compare the two, you can see some differences. There's a little bit of difference in the peak width from both samples, but more interestingly, we see that the average mass of the Sirion particles is about 2.8, and the average mass of our ViroVec samples are about 3.7. So these are a bit lighter compared to the Sirion samples. Now, if you would theoretically calculate the mass of an empty AAV8 particle with a VP ratio, so you have these three capsid proteins, VP1, VP2, VP3, but you would use a more conventional theoretical ratio of 5 VP1s, 5 VP2s, and 50 VP3s, you could calculate a theoretical mass of about 3.7. But clearly the Sirion sample here is not the same. It looks a bit heavier. So together with our collaborators at Roche, we performed a different assay, a capillary electrophoresis assay, where we would take our pools of AAV, both from Sirion and ViroVec, and separate the VPs, the VP1, VP2, and VP3 proteins, and basically quantify the amount of VP1, VP2, and VP3 in those pools of AAVs. All right, so for our AAV8 sample, if we look at the ViroVec, they contain about, on average, 4 VP1s, 5.5 VP2, and about 50.5 VP3 proteins. And this would theoretically give a mass of about 3.7 megaton. If we look at the serial sample, we have more VP1s, about 8.2, more VP2, 14.5, and less VP3, 37 VP3s, which gives a theoretical mass of about 3.86 megaton. So this nicely coincides with what we saw in our mass analysis assay. So with the mass photometry and CDMS, that the serial samples are a little bit heavier, and that could very well be because of the different ratio of VP1, VP2, and VP3 proteins in the capsid. Okay, so we repeated the whole assay, all the mass analysis, and also the capillary electrophoresis as well, but this time for a AAV2 serotype. So again, if we look at the mass analysis, if we look at the serial sample, mass photometry gives us a mass of 3.8, and CDMS again around 3.8 as well for our AAV2 particles. ViroVec one are again a little bit lighter, 3.7 and also 3.7 with the CDMS. So if we look at the VP ratio there, can we see again, explain these differences between the masses of these serial and ViroVec samples? Yes, indeed. If we look at the ViroVec samples and determine the average VP1 and VP2, VP3 particles, we can see that ViroVec has about 6 VP1s, 4.5 VP2, and 49 VP3 particles, which gives us a theoretical mass of about 3.76. And the serial again carries more VP1, about 8.5, and also more VP2, almost 13, and less VP3. So in the end, there's indeed a higher theoretical mass for that, around 3.87. So again, this nicely coincides with each other. Okay. Yeah. So far, we've only looked at empty particles. So these are AAVs that do not carry any transgene. But what if we have AAV particles that do carry a transgene and are purified following filling of the capsid with a transgene? So for instance, here we have an AAV8 particle, Adeno-associated, but this time a transgene is packaged, which contains a CMV promoter and a GFP gene, and altogether it's about 1 megadalton. So we can do again our CDMS assay. This was a previous result looking at the empties, but if we now look at these particles which contain a transgene, we see a shift indeed. So this is the 2D histogram of our AAV particles, and you can see that in the M over Z, in the mass spectrometer, indeed they are shifted to the right. In intensity, they are similar to the empties. So basically, they carry the same amount of charges, but they just are heavier. They just contain more weight, which makes sense. So in the mass histograms, we can see this as well. We see a population here, a main population, which has about a mass of 4.9 megadalton. And there's also still a little bit of particles left that are not containing a transgene, which are basically empty. And indeed, if you look at the empties, then we can say, yeah, indeed, about 1 megadalton is packaged in these filled particles. Okay. How about the ViroVec samples? Also there, we looked at particles that contain a transgene. Also here, a transgene with a CMV promoter and a GFP, but the transgene is designed slightly different, so they're a bit lighter. They're 0.7 megadalton. And if we then perform again the CDMS assay, we get a result like this. So a main population containing the transgene, which is about 4.6 megadalton, but also a population that carries more weight. So they're overfilled or they're packaging extra material, which is about 5.1 megadalton, and a slight amount of empties as well. All right, we at this time thought using CDMS to analyze these particles can be quite harsh. So as you saw earlier, particles have to go through vacuum in an electric current, go to the orbit web where it spins around. So what is there potentially bias that we create by using these more harsh conditions? So again, we used mass photometry. This time we can nicely compare the same data format CDMS, but also with mass photometry. And here you can see, so I put them one next to each other. So this is AAV8 filled particles from Sirion and from Viruvec. And the CDMS we saw just before, but the mass photometry data actually nicely shows the same image. So the data is really in agreement with each other. Again, a main distribution of filled particles for the Sirion sample, also around 5 megadalton. And for the Viruvec, these three populations with the main population of filled particle containing a single transgene, slightly overfilled population, and a very small amount of empties. So this was for the AAV8 serotype. Of course, I repeated everything, but now also for the other serotype, the AAV2 serotype. And again, here we can see that CDMS and mass photometry really show a similar image. So in the CDMS, we can see for the Viruvec or the Sirion sample, sorry, a main population of filled particles containing a single transgene, a small subpopulation containing partially filled AAVs, and a small subpopulation containing empty particles. And in the mass photometry, you can actually see the same image. So most of them show filled particles with a single transgene, partially filled, and empties as well. And if we look at the Viruvec samples, we can again see in the CDMS that most of the particles contain just a single transgene, but also a small amount contains extra material. And with the mass photometry, we basically see the same image. All right, so perhaps a small recap of both techniques that we show here. CDMS, it's a mass spectrometry based technique. So it's in the gas phase. It takes about 10 to 30 minutes to do a measurement. And we've used an orbit trap here, commercially available mass spectrometer. However, it would require probably some expertise in order to tune the machine in such a way that the AAVs nicely goes towards the mass analyzer, and also the data afterwards might require some expertise to analyze. On the other hand, we have the mass photometry, so it's a solution based technique. We just simply dilute our AAVs in PBS. Measurement takes about 5 to 10 minutes because you first need to calibrate the machine. And here I've used a commercially available mass photometer, SAMOX MP by Refine. And the data analysis is also quite straightforward. So in conclusion, both techniques, CDMS, mass photometry, they're quite useful and are accurate and fast in determining the mass of AAVs. Different production platforms of the AAVs also show differences in the end. So we saw differences between the mammalian cells and the insect cell AAVs, and both the CDMS and mass photometry are in nice agreement. So that's really good to see. So with that, I would like to also end my talk, and I'd like to thank my group members, especially Victor and Sushui and Ayan for helping me measuring CDMS and mass photometry. And also Albert, of course, for giving me a place in the lab and to let me work on this exciting project. And our collaborators, Alisa, Marcus, and Marco, for all their help in this project. And I would like to thank you for listening.